Thank you for the invitation. I'm really honored to be here. What I'm intending to do is what means safeguarding the gene pool of Artemia and review what means characterizing and monitoring Artemia genetic resources. These are two goals of this meeting. Let me explain why a guy from far away from Artemia production is doing here. Maybe you don't know Chile. Chile is a long country, narrow in South America, as you see in the map. And it happens that we have two of the oldest Artemia species, Artemia franciscana and Artemia persimilis. Franciscana is in the north, the Atacama Desert. That is the southern edge of Artemia franciscana distribution. And maybe you know that species at the edge could have very unique populations. So that's why it's important to see what happens in Chile. And also we have a really strange situation because we have subantarctic lagoons in Patagonia. Look, the latitude 52, and in the north is 27. So Artemia persimilis lives there. We don't know how Artemia persimilis arrived there, but it's really interesting. And also we have a special kind of problems, different of what we have seen here. Chile is one of the main lithium producers, and we think that pumping shrimp, brines from beneath the solar is maybe affecting the lagoons, the salty lagoons. Yeah, what I'm talking about is the International Biodiversity Framework. The components of Artemia biodiversity, genetic variability, as Graham mentioned, population. What is important from populations is that retain local adaptations. And species, well, in Artemia we have sexual species and also parthenogenetic lines. And ecosystems. Well, ecosystems contain regionally endemic species, but are, as we saw in Great Salt Lake, are shrinking at alarming rates. For example, Urmia Lake, Great Salt Lake, others. I would like to propose a kind of pragmatic approach to see our role as the consortium, which means that safeguarding gene pools and the adaptive potential of Artemia, of wild and domestic or farm types following Aquaculture. This requires joint collective effort to comply with international guidelines, to understand that genetic variability fuels population species resilience and adaptive change. So we need to monitor Artemia populations or stocks. It's important to share conceptual background. For example, what a population, stock, farm type and species is. Clear goals, how genetic diversity and adaptive traits distribute within a species. That is something that we need to work on. And the other aspect is to have a cost-effective tools. Despite multiple markers available, we should use only those reliable and cost-effective. This is because we need to compare data from different regions. What is the international framework? Well, there is limited information below species level. This is called the hidden biodiversity crisis. We don't know much about Artemia populations, stocks around the world. The other international framework is that set in Montreal in 2022 that says that the genetic diversity within and between populations of wild and domesticated species is maintained, safeguarding their adaptive potential. And they propose there 3 biodiversity indicators. Graham mentioned about it, but I think it's important to consider in Artemia. First, identify and maintain genetically distinct or isolated populations. Well, stocks, sensu aquagris. The size of the population should be important. I don't think this is really a problem in Artemia, so I will skip that. And the number of species in which genetic diversity has been or is being monitored using DNA-based tools for at least one population. I don't think we are doing that. Well, the other international context is the Sustainable Development Goals. So the challenge for this group is to assess and monitor the genetic diversity within species, what we call interspecific diversity, of wild and domestic species. We call it farm types. Identify and maintain and monitor genetically distinct populations. Reduce, this is, I think, is very important, the introduction of invasive species. A lot of people we saw yesterday are moving Artemia from one place to another. According to the Montreal conclusions, we should reduce by 50% invasive species and manage them to minimize their impact. So how we could translate this information into policy? I found yesterday very important what you are doing in Great Salt Lake. So, for example, what we could do to avoid the shrinking of Artemia lakes. Monitor is very important. So we ask how climate change and human intervention will affect ecosystem services. There are very important services of Artemia lakes. For example, wild diversity, cyst production, that is very important for this meeting, etc. Or avoiding local population loss. Local population is very important. I mean, in evolutionary terms, it's the first step toward speciation, but also it's very important to keep these local populations. Gene level biodiversity. This is the ultimate source of variation for evolution and adaptation. We have 2 or more Artemia genomes. The size is very important. The number of genes, as you see there, it's similar to the human genome, so this is interesting. Yeah. And if you check in GenBank, you have more or less 19,000 protein coding genes sequenced. So that is really important. And all these genes are distributed in 21 autosomes and 6 chromosomes. And this is something very important in Artemia. About more than 60% of the genome is repetitive DNA. Wow, that is amazing. So we need to look at that. So here below you see the published genomes. I know there is another one circulating in some journals. So what is the message to take away from this part is that Artemia obviously is the most studied species. The genome available will facilitate a lot of problems that we need to address, yeah, and will allow to characterize the sex chromosomes. That is something that a group in Austria is doing it, the one on the right. Also, we have mitogenomes, and I'm really impressed to see that Artemia franciscana, the mitochondrial genome is sequenced, Artemia sinica, Artemia urmiana, Artemia persimilis. This is done by Lejean's group in persimilis. Hey, what are you doing so south? No, but it's important information. And Artemia salina. You see there the number of genes in this small kind of DNA, but it's important to have this information. So, in short, we have multiple and variable nuclear and mitochondrial genes to work with for different purposes. If we talk about the topic of cyst production, I think this is a very important adaptive trait. So we have seen only environmental aspects, but there are genetics and also epigenetics, yeah, and interaction between genes and environment. So cyst production is a very complex thing. And cysts are important because they contain a genetic memory of the past. And this is why cyst banks are really important. So, for example, there you have the numbers of genes and proteins involving in different aspects of cyst production For example, 300 protein coding genes control cis diapause, control high salinity and anoxia tolerance. So a lot of genes. Also for desiccation, tolerance, UV, temperature, fluctuations, and dry environments are protected by different kind of genes and proteins. And also we have single genes, and this is very important. We have a different kind of genes, and I wanted to highlight here gene nanos. Dr. Han did this work with Ligin, and I think this is a very important achievement. If you knock down this gene, I mean interferon RNA transcription, Artemia start producing cysts. Yeah, so I think you should continue along this line. It's very important. So you have many other single genes. Well, population biodiversity. Well, have a look to this number 129 locations reported by Gilbert van Stappen in 2002 in the Artemia book. We have 73 sites in South America, Argentina, Bolivia, Marcelo is here, Brazil, Chile, Colombia, Ecuador, Peru, Venezuela. And I brought this study because I like the map. So you can see that Munoz did a characterization of, he says, the entire native range of Artemia franciscana. He compared 39 sites, and he found 12 genetically different for 2 genes. Yeah? So that means that 12 out of 179 are known. The others, what is happening to them? These are local, genetically distinct, allopathic, that means isolated populations. So these are the, according to Aquaculture, stocks. Yeah? From my knowledge, none of these are regularly monitored with DNA-based methods or tools. How many of the 179 reported still exist? It's a very good question. Well, that is important. In this case, they found that bird dispersal was not important, at least for these 12 populations. We did something similar using a very distinctive trait of Artemia franciscana, that is ALU sequences, that is part of this repetitive DNA. So as you see in that figure, you can establish a difference between Artemia franciscana and Artemia persimilis in the upper part of the figure. Artemia persimilis doesn't have these sequences. Artemia franciscana, it does have it. And I think it's very important for Artemia franciscana plasticity to have these sequences. So you find it, introduce it in different parts, maybe because of this ability of these sequences to transposed along the genome. 60% of the genome is repetitive DNA. So other Artemia species, numbers means location listed by Stappen. In Europe, 145 Artemia sites, 12 countries. Tarquinia, the place you are going on Thursday, has Artemia salina, maybe? Had. Wow. Thank you. So what do they have now? Okay. So what did you find, Artemia salina?
I think so, yes.
Okay, great. Okay, but this means that it's very important to monitor populations. In Asia, Gilbert found the number you see there in 16 countries. In China, 88 sites, 12 in Iran, yeah. And so the question remains, how many genetically distinct populations identified within species or stocks? Yeah, I know that Li Jin is going to talk about this, yeah. How many of the original samples are maintained, yeah? How many being monitored with DNA-based genetic diversity? I'm not aware of any. Yeah, this is critical and sad. Yeah, except for Assem, who did a study in Lake Urmia for a long time, but taking cyst. Yeah. I don't think that cysts represent the whole population, but we need to discuss about this. Okay, new sample locations, 46 lakes sample in Siberia between 2002 and 2003. Lyudmila is involved in this work. Alireza Assem found 59 new sites in Urmia, in Iran. This is recently published in So Diversity. 3 lakes in Crimea. This is done by another group. I mean, Elena, Nikolai, yeah, yes. And 36 lakes studied 56 samples in Russia and Aral Sea, yeah. Boyko is going to talk about this, I guess, this meeting. About monitoring farm types, okay? We think that the Vinh Chao strain, it's a farm type, but I don't know what is happening with this strain. Is it being monitored, kept somewhere? Yeah, question for the audience and for what? Okay. This is very important. This is an example of the importance of monitoring and the importance of have a cyst bank. New species discovered from hatched cyst. And I put this information from the Miami Herald. A journalist described this, and he says something like this: the extinct creature has risen from dead. Well, sort of. Yeah, so you can see there. The samples were preserved at Torre de la Sal in Spain, by Francisco Ontoria. I should have a minute to inform that he is very ill, Francisco. And new species were discovered from this cyst. And no new record of the species were documented in 30 years, monitoring. They existed only in a cyst collection. So the species are Artemia amati, new species, and Artemia sorgelusi. Yes, he says, the journalist, new species named after professor who contributed a lot to the study of Artemia. So congrats, Patrick. And what is needed is further research about these new species. So, regarding species diversity, we have these numbers. We have 3 species in the Americas. We consider Artemia monica as a species, really species. We can discuss this later. So we have 4 species in the Old World, Artemia salina, Artemia urmiana, Artemia sinica, Artemia amati in Kazakhstan, and 2 species from the Tibetan plateau, Artemia tibetana, Artemia sorgelosi. Well, no, I didn't want to show that. Sorry. Well, my conclusions are genetic diversity indicators are a complementary effort to report the genetic status of a species, but allows tracking, reporting, prioritization, and communication about this group. I think is important. The collective role of this group is to comply with the CBD guidelines, specifically with biodiversity indicators. The reality for Artemia, as well as other species, is the lack of information below the species level. We need to identify and maintain genetically distinct populations. We need monitoring populations with DNA-based and cost-effective tools, markers, and the example of new species discovered from cysts show the importance of monitoring. And finally, I think this is an old version, but I think Aquaculture is a very important tool for doing this monitoring. So I appreciate the presentation of Graham, and we need to follow Aquaculture for have a very good description of Artemia intraspecific biodiversity. Thank you.